Mycoplasma. In this video, we will discuss about mycoplasma, structure of mycoplasma, classification of mycoplasma, sensitivity of mycoplasma, nature of mycoplasma, reproduction, and mycoplasma and diseases. Mycoplasma are the smallest non-prokaryotic organisms characterized by the absence of cell wall. These organisms were discovered in 1843 by Pasteur in the pleural fluid of cattle suffering from pleuropneumonia and were therefore called pleuropneumonia-like organisms. It's also called MLO, that means mycoplasma-like organism. The credit for growing these organisms in cultured medium goes to Nocard and Rocks in 1898. They also demonstrated that if healthy cattle were injected with these microorganisms, the symptoms of disease appear. Later, Novak in 1929 placed them in the genus Mycoplasma. Mycoplasma are now known to infect a large number of animals. For example, dog, sheep, mice, and even man. And plants, for example, potato, corn, brinjal, etc. Occurrence. In nature, they occur in soil, sewage water, and in plant and animal bodies. Besides, they have also been found in hot water springs. In the laboratory, they are common contaminants of cultures rich in organic matter. Structure of mycoplasma. Mycoplasma are unicellular prokaryotic organisms. They are smaller than viruses. In fact, Mycoplasma ledlii has been listed as the smallest of all the free-living organisms with a diameter of 0.1 to 0.3 micrometer. They do not have a cell wall. The outermost limiting layer of the protoplast is a cell membrane which has a typical trilamellar unit membrane structure. Absence of cell wall makes these organisms highly plastic. Therefore, these can assume various shapes such as spherical, granular, filamentous, cocoid, etc. So, they are pleomorphic. The cells are non-motile, but a few forms show gliding movement. Cell membrane encloses protoplasmic matrix, which contains ribosomes, fatty substances, and proteins. Proteins represent as many as 40 enzymes, which are concerned with the metabolic activities of the cell. Protoplasm of mycoplasma has both RNA and DNA. RNA is single-stranded and occurs both in the ribosomes and cytoplasm. DNA is long, coiled, double-stranded thread that extends almost throughout the cell. The amount of RNA and DNA in these organisms is less than half that is usually present in other prokaryotes. In some species, for example, Mycoplasma gallisepticum, polar bodies called glebe protrude from one or the other end of the cell. These are considered to be the sites of enzymatic activities and attachment during infection. A replicating disc is present at one end which assists duplication process. The mode of nutrition in mycoplasma is heterotrophic. Classification of mycoplasmas. The following three genera of mycoplasma have been recognized on the basis of their nutritional requirements. 1. Mycoplasma, second, Acoleplasma, and the third is Thermoplasma. First, Mycoplasma. These mycoplasmas require cholesterol for their growth, which is incorporated in their cell membrane. They are parasitic and infect mucous membranes and joints of humans and animals. So the second is Acoleplasma. Acoleplasma, these mycoplasmas do not require cholesterol for their growth, but will incorporate it into the membrane. If it is furnished in the medium, they are found in vertebrates and sometimes in plants, and also occur in soil and sewage water. Third is Thermoplasma. These mycoplasmas also do not require cholesterol for their growth. They are strictly aerobic, acid-loving microorganism growing well at pH 0.96 to 3.0. The optimum temperature for their growth is 59 degrees centigrade. Now the sensitivity of mycoplasmas. Mycoplasmas are gram-negative. They are insensitive to penicillin, but sensitive to other antibiotics like streptomycin, erythromycin, chloramphenicol, etc. Their insensitivity to penicillin is perhaps due to the absence of cell wall. This antibiotic interferes in the synthesis of peptidoglycan, an important constituent of the bacterial cell wall. Nature of mycoplasmas. Mycoplasmas can pass through bacteriological filters and do not possess a typical bacterial cell wall. These two characters confirm that mycoplasma are not bacteria, as these microorganisms can multiply in medium which does not contain living tissue. Hence, they are not viruses either. Viruses show characters of life only inside the living host. These characters therefore show that mycoplasmas are intermediate between bacteria and viruses. Cultivation of pathogenic mycoplasmas. Pathogenic mycoplasmas can be cultivated on medium containing proteins, sterols, phospholipids, mucins, and nucleic acids. That means DNA and RNA. The growth of some obligate anaerobic mycoplasmas is stimulated by adding yeast extract and cholesterol to the medium. The optimal temperature for their growth is 36 to 37 degrees centigrade. On a solid medium, the colonies of mycoplasmas have a characteristic round form with a thickened center and a delicate periphery like a fried egg. Aerobic mycoplasmas show on a medium supplemented with 20% horse serum. Now, reproduction. Not much is known about the reproduction of Mycoplasma. However, four types of cellular bodies of Mycoplasmas laid lowi have been found. These are elementary bodies, secondary bodies, tertiary cell bodies, and quaternary bodies. Elementary bodies reproduce by a process similar to budding. These grow in size and then again give rise to elementary bodies. A process similar to binary fission is also said to occur. Some important mycoplasmal disease. Mycoplasmas cause serious disease in human beings, animals, and plants. Some of these are described below. Plant diseases. Many plant diseases caused by Mycoplasma were earlier considered to be viral diseases. The symptoms of diseases caused by viruses and Mycoplasma are similar and it is difficult to distinguish between them. Diseases caused by Mycoplasma are generally transmitted through insects, such as leafhoppers, mites, and flies. Some of the common non-plant diseases are little leaf of brinjal, bunchy top of papaya, potato witch's broom, mulberry dwarf, maize stunt, and clover dwarf. Animal diseases. Pathogenic mycoplasmas affect the respiratory organs, central nervous system, cardiovascular system, and urogenital system in human beings. Mycoplasma hominis, a species studied most extensively, causes pleuropneumonia, inflammation of genitals, non-specific urethritis, prostatitis, endocarditis, and other diseases. Mycoplasmas generally produce diseases in man when his body resistance is significantly low. Mycoplasma pneumoniae causes primary atypical pneumonia, PAP, hemorrhagic laryngitis, and vesicular inflammation of tympanic membrane. Mycoplasma hominis and Mycoplasma fermentans are known to cause infertility in men. Mycoplasma mycoids cause pneumonia in cattle. Thank you.